The early data is very encouraging, and I think it gives promise for what the direction that we're going to be moving in with this product. Hopefully, we're entering into a new era of medicine where we can harness the immune system, harness the power of the immune system in order to attack the cancer.
Welcome back, everyone, and thank you for joining us for another Virtual Investor KOL Connect segment. Today, we are featuring Senti BioSciences and are very pleased to have Dr. Kanya Rajangam. She is President, Head of Research and Development, and Chief Medical Officer at Senti, as well as Dr. Stephen Strickland. He is an internationally respected leukemia researcher and Director of Leukemia Research for the Sarah Cannon Research Institute. Welcome, and we appreciate your time today. Before we get started, I just want to remind our audience that Senti BioSciences is publicly listed on NASDAQ and trades under the ticker SNTI. And during today's discussion, the company will be making forward-looking statements, and I encourage everyone to view the company's latest SEC filings on their website at sentibio.com for the latest information. Okay, to start, Dr. Strickland, can you give us a brief overview of acute myeloid leukemia, also known as AML, including the current treatment landscape and some of the key unmet needs that persist for patients today?
Sure. Thank you for having me and for giving me the opportunity. But AML is a challenging diagnosis to deal with amongst the hematology oncology community. Basically, what it is the bone marrow leads to an inappropriate overproduction and subsequent accumulation of immature cells, or what we call blast cells. And these blast cells infiltrate the marrow and they disrupt the normal production of our normal white cells, our normal immune system, as well as the red cells and platelets. And so patients can be at increased risk for life-threatening complications such as infection or bleeding. It is, if left untreated, a terminal diagnosis. And so we tend to have to treat it aggressively and often very quickly upon its discovery. It's also a diagnosis which historically has been treated with intensive chemotherapy ever since the early 1970s. And for many years, that's really the only treatment opportunity that we had. That led to many challenges for our older patients, especially when the median age of this diagnosis often hovers between about 67 to 70 years of age. So as you can imagine, many of our patients may not be candidates for intensive therapy due to their age or other comorbidities that tend to develop in that older age population. So we need desperately additional therapies. And although there have been improvements over the past several years with some lower intensity options, unfortunately, for the majority of our patients, the current treatments that we have today, for many, are not curative. And in that regard, we need better and more effective therapies to either help patients get into better remission or help patients who have relapsed achieve remission and hopefully a cure. But that's some of the historical challenges that we have, and also the fact that many of these patients ultimately have to undergo hematopoietic stem cell transplantation to give them a possibility of a cure, which comes along with the potential for cure, but also with potential morbidity and potential mortality relative to that procedure.
Dr. Rajangam, for those less familiar, could you provide a high-level introduction of Senti 202 and the key differentiators that make this approach unique within the broader AML pipeline?
So, Senti 202 is a first-in-class logic-gated selective anti-AML off-the-shelf CAR-NK cell therapy. What we did is we designed Senti 202 to address the key challenges in developing new therapies for AML. We did that by inserting what we call a gene circuit into a healthy natural killer cell to augment its intrinsic leukemia-killing abilities. This gene circuit has 3 proteins. The first protein is a kill protein, which we call the OR logic gate. Basically, what that does is it addresses the heterogeneity of AML cells by killing cells that express either CD33 or FLT3. That's important because between these two, nearly 95% of AML patients tumor express one or the other of these 2 proteins. And furthermore, CD33 is found on what is known as bulk AML blasts, and FLT3 on these niche small cell population called leukemia stem cells, which are known to drive shorter periods of remission and repopulate the bone marrow with AML after patient has initially responded. So it's important for us to kill both the bulk blasts as well as these leukemia stem cells. The second protein we have in our gene circuit is what we call our NOT logic gate. This has a protect function, and what it does is, since AML targets, including CD33 and FLT3, are also found on healthy hematopoietic stem cells, this NOT logic gate protects the healthy cells from potential off-tumor on-target effects. The way it does it is by recognizing this protein called endomucin, which is found on the surface of healthy hematopoietic stem cells. And when Senti 202 recognizes endomucin, it doesn't kill the cell, even if it expresses CD33 FLT3. By this way, we've been able to engineer in the selectivity so that Senti 202 kills leukemia cells and protects the healthy cells. The third protein we have enhances the activity of both Senti 202 and host immune cell by incorporating a cytokine which is needed to support the function of these cells.
Dr. Strickland, from a clinical perspective, what has stood out to you most in the data generated to date with Senti 202, both preclinical and any early clinical insights?
Sure. I think from a preclinical perspective, something that's intriguing to me is obviously the design of this product. It's an allogeneic cellular therapy product, so it's someone has donated their cells, and their immune cells have been generated in a manner in which to hopefully seek, attack, and destroy a property which we know exists on many of the acute leukemia cells. Actually, two different properties which can exist on the leukemia cells, both CD33 as well as FLT3. And so by doing so, hopefully we're entering into a new era of medicine where we can harness the immune system, harness the power of the immune system in order to attack the cancer. We know that our native immune systems often are suppressed in a way that are not able to attack the cancer cells, and that the cancer, when it's developing, finds way to evade our own normal immune systems. And so taking immune cells from someone else, engineering them in a manner to have a validated target that they can go after to have anti-cancer activity is very intriguing. When it comes to the clinical aspect of this, I would say that early on, what we're seeing is no significant dose-limiting toxicity. We're seeing activity, we're seeing responses, but we're not increasing the toxicity, which has been sort of one of the aspects which has plagued drug development, therapy development for quite some time. And so if we can find a way to more effectively target the cancer without increasing toxicity and with improving overall activity, we're hopefully going to be entering into the sacred territory of better therapy for our AML patients.
Appreciate that. And so to build on that, how did you envision Senti 202 fitting into the AML treatment paradigm and potentially changing the standard of care if ongoing development is successful?
Sure. Well, the first thing we need, obviously, to change the standard is an effective therapy. Obviously, the early data is very encouraging, and I think it gives promise for what the direction that we're going to be moving in with this product. I think that what we really need is more effective therapy. We know that many of our patients, unfortunately, relapse, even quite a few of those who are taking the stem cell transplant. And so more effective therapy to get these patients back into remission. If we're able to do that, able to harness the power of the immune system to attack the cancer and hopefully lead to deeper, more durable remissions for these patients, especially in the relapse refractory setting, then the next step will be, will it be beneficial to actually move a therapy like this into the frontline setting, possibly as a consolidative approach after patients are in remission. Once they achieve that remission, and instead of maybe taking them to transplant, can we give them a shorter course, sort of immune therapy, cellular therapy that would hopefully extend the duration and optimize the duration of that remission, hopefully cure. Can we do so with a therapy that then wouldn't have the morbidity of a transplant, something like graft versus host disease or or the toxicity of the conditioning therapy that many of the patients experience. So can we achieve a more effective therapy, a therapy that will lead to better, deeper remissions, and hopefully more cures, which is ultimately what we're striving for.
So, looking ahead, Dr. Rajangam, what are the key development milestones and clinical trial goals that investors should watch for over the next 6 to 12 months?
So first up, later this month, we have 3 presentations at AACR. We have an ongoing phase 1 trial with Senti 202 that has been accepted for an oral presentation. We are very excited to share our initial clinical data with a broader group of folks. We also have, in addition, correlative work that is going to be presented at AACR as well. What we've done, we're working in collaboration with scientists at MD Anderson, is look in great detail as to what happens within the bone marrow of patients who are exposed to Senti 202, can compare it to baseline. So we are really excited to be presenting that data as well at AACR. And finally, we have a preclinical poster that gives additional mechanism of action support to our unique approach with the logic-gated gene circuit design. Beyond that, we are on track to have about 20 patients or so by end of the year, and we hope to continue to see these responses and the durability with larger numbers of patients. In parallel, internally, we've started ramping up our CMC efforts to get ready for future pivotal trials, assuming the data continues to support that evaluation.
Well, certainly an exciting time for Senti Bio. And with that, this concludes our Virtual Investor KOL Connect segment featuring Senti BioSciences. I'd like to thank Dr. Rajangam and Dr. Strickland for their time today. As a reminder, Senti Bio trades on NASDAQ under the ticker SNTI. And if you like what you saw today, I encourage you to visit sentibio.com for more information on the company, to follow their social channels, to stay current on the latest information. You can also visit virtualinvestorco.com for the latest events calendar and segments. I wish everyone a great rest of your day.